This is going to be Chapter 9, the General and Special Senses, Part 2. This is going to be 4.0 Vision. We're going to talk about accessory structures, the eye, anoscoria, hyphema, cataracts, visual physiology, visual acuity, accommodation problems, the visual pathways, night blindness, and eye emergencies. 4.1, the accessory structures of the eye. Eyelids and associated exocrine glands in this section. Superficial epithelium of the eye, tear secretion, and extrinsic muscles. 4.2, the eyelid, palpebra, they act like windshield wipers. Bleaching keeps the surface of the eye lubricated. Medial and lateral canthus, this is where eyelids are connected at these locations. And 4.4 is a lacrimal caruncle, soft mass of tissue that produces thick secretions, and this is why you get matter in your eyes in the morning whenever you wake up. 4.5 is conjunctiva, epithelial layer that covers the inner surface of the eyelids and the outer surface of the eye. Asty is an infection in a sebaceous, tarsal, or sweat gland of one of the eyelashes. Conjunctivitis, pink eye, results in damage to and irritation of the conjunctival space. 4.6 is the lacrimal apparatus that produces and distributes and removes tears. 4.7, lacrimal gland, provides key ingredients for most of the volume of tears. Dozens of empty pockets between the eyelid and the eye. Pardon me. And these are going to be the eyelashid, palpebra, which is here, medial canthus, and this is going to be the lateral canthus. Obviously, sclera, or the white of the eye, the iris, or the color part, and then the pupil. You have lacrimal glands, noted here, and these will create tears that keep and moisten the conjunctiva. Also, you have lacrimal sacs, inferior lacrimal canal, nasal lacrimal duct. Some people, this is actually open to where if they blow into this, they can protrude their eyes. If you're interested in that, you can look on Ripley's Believe It or Not. Extrinsic eye muscles, figure 9.10. This is what allows for lateral movement, up and down movement, and pretty much movement of the eyeball. You have lateral rectus, medial rectus here, superior oblique, superior rectus, and inferior rectus, inferior oblique muscles. And this allows for turning of the eyeball, the maneuvering left and right, and obviously up and down. 4.11, the eyeball is hollow, and you have essentially two cavities in this. You have a posterior cavity, which houses vitreous chamber, and you have an anterior cavity. And in the anterior cavity, you have actually two cavities. So again, the eyeball, I'll kind of draw it on here. And then you have a front portion of this, or anterior cavity, and then this anterior cavity actually has two more cavities in it, an anterior and posterior chamber. And if we'll take a look at this one here, first of all, let's take a look here. Anterior cavity and a posterior cavity, and from those actually have an anterior and posterior chamber in this one, and then this is where all the magic happens, to where light and visual information comes in, and it's assessed on this back lining. We can see down here the posterior cavity, the vitreous chamber. This is all of this area. And then you have coverings to this. There's fluid in here. And as visual information comes in, it'll actually seat on the back of this, and all this area here would be information that would be nerves or sensory input to to the optic nerve. In the front chamber, the anterior cavity, you have two chambers, posterior and then an anterior chamber. So take a look over those structures and please be aware of that. 4.13, fibrous tunic. Fibrous tunic consists of the sclera and the cornea. The sclera is the white of the eye, and the white of the eye is connective tissue, and the cornea, continuous with the sclera. Collagen fibers are organized into a series of layers that do not interfere with the passage of light. 4.14, vascular tunica. Vascular tunic provides a route for blood vessels, regulates the amount of light, secrete the aqueous humor, control the shape of the lens, and consist of the iris, ciliary body, and choroid. And 4.15, the vascular tunic contains the iris, ciliary bodies, and supensitory ligaments, which adjust the pupil. Now this is going to allow various amounts of light to come in. So this is what we were talking about right here. This is light coming in. This is the cornea, or the surface of this, conjunctiva. And then you have, let's see, the vascular tunic, which is here, which is the iris, or the opening, the ciliary body, and this is going to adjust the lens, and then the choroid, which will supply blood vessels and nutrients for the posterior cavity, also assist in producing the vitreous humor, which is in here. Figure 9.12, pupillary muscles, and this is what makes our eyes adjust to light. This would be the picture you would see if someone was under the effects of a narcotic, which would be termed as dots on a road map, or pinpoint pupils. These are dilated over here, very large. This person probably got dilated for visual acuities in an ophthalmologist, but this would be the picture if someone was on an amphetamine that you would see. So amphetamines like cocaine, methamphetamine, they're going to give you a dilated pupil. Things like narcotics are going to give you pinpoint pupils. Clinical notes, aniscoria, which pretty much means unequal pupils. And these are pretty much controlled by the ocular motor nerve. In the event of a brain lesion, intracranial pressure increases and your actual pupils are going to dilate on the affected side. So an example of this, let's say I have a right brain injury from intracranial pressure or brain lesion, my right pupil is going to dilate. 4.19, the neural tunic. Neural tunic or retina, two parts, and we have pigment and neural. Pigmented absorb light after it passes through the neural part. The neural part, photoreceptors that respond to light supporting cells, neurons, and blood vessels. 4.20, the retina. We have several terms here. Rods, which are photoreceptors for night vision, and cones, which are for your color vision. The macula lutea, concentration of cones, and this would be a concentration of color vision or receptors of color vision. Fovea, highest concentration of cones in the macular lutea. Visual axis, imaginary line from the center of an object through the center of the lens. Bipolar cells, rods and cones, synapses through 6 million bipolar cells. And a bipolar cell is a type of neuron that are special sensory, if you'll remember from the last chapter. Ganglion cells, bipolar cells, synapses with the ganglion cells. And we'll take a look at this here. So this is where light would come in. These are the ganglion cells here. And this area here is the bipolar cells. And those bipolar cells take the rods and the cones. And then this here is your pigmented area. So rods and cones is where you're going to get your night vision from this area here, and your color vision. And this is going to be sensory input from here and here from this massive area that's on the inside of the eye. So as we look at this, this is vascular supply here. Pigmented part of the retina is in this location. Neural part of the retina. And this is going to be considered the optic disc. And this is a picture of a real one here. This is the fovea. And then this is the macula lutea here. 4.22 chambers of the eye. You have the anterior cavity. This is the front part, and this actually has two chambers in it. This has an anterior and a posterior chamber. The anterior chamber extends from the cornea to the iris. The posterior chamber between the iris' ciliary body and the lens. This is filled with aqueous humor. So in the anterior cavity, it is filled with aqueous humor. The posterior cavity contains aqueous humor known as the vitreous body and contains vitreous humor. Vitreous humor maintains the shape of the eyeball and holds the retina against the choroid. Glaucoma, which is a disease, interferes with the normal circulation and reabsorption of aqueous humor, and an elevation in the pressures of the eyeball can actually cause blindness. Alright, in the anterior cavity we have the anterior and posterior chamber, and the posterior chamber, and in the posterior cavity we have the vitreous chamber, which is back here. Again, choroid, retina, sclera, the white of the eye, ciliary body. This is going to adjust the lens, body of the iris, maybe the colored part of your eyeball. Alright, figure 9.14 or 914. This is if you actually print this out, you can take a look at where your blind spot is. Now the goal of this is to actually stare at either the plus or the dot and pull it closer to you. And we're do it in a very slow fashion, and one of these will disappear. But what is happening is this one that disappears, if you're looking at this one, is going to travel across where all of your nerves and where all of your blood vessels are at the back of your retina, and this is considered your blind spot. 4.25, hyphema, is the presence of blood in the anterior chamber of the eyeball, and this is sight threatening. If you get blood and it clots up in there, you get solid substance that's not allowing light to enter into the iris or the lens. 4.26, the lens lies posterior to the cornea, held in place by the suppensatory ligaments. Primary function of the lens is to focus the visual image on the photoreceptors, and these are going to be on the back of the eye in the posterior chamber. It does so by changing the shape of the actual lens. And it depends on the amount of focal distance here. So let's say that this is the back of the eye, and what's going to happen is the lens is going to change its shape so that it focuses the image onto the back of the eye. A close source would increase the focal distance. Medium source, again, the lens changes shape. So ciliary muscles contract the lens around for close vision and flatten for distant vision. So it adjusts the shape of the lens depending on how far or how close the object is. 4.29, clinical note on this. This is cataracts. Lens loses its transparency with age, and caused by drug interactions. Radiation can also cause it. Senile cataracts are caused by age and yellowing of the actual lens, corrected by replacement of the lens of the eye. Refractation, accommodation, and image formation. Three terms here. Refracted, Light is bent from one density to another. And then the next term is going to be focal point, a point at which the light rays converge. And then last but not least is going to be accommodation. And this is going to be changing in the shape of the lens is what accommodation means. So again, same picture. And we just went over this. So if the target is close, lens changes and is pretty much rounded. And if the object is far away, the lens flattens for distance. Image formation, and everything is kind of upside down and backwards as it's imaged onto the eyeball. If you'll notice, this is the top and this is the actual bottom. This was on the right as we were looking at it. Now it's on the left as it's entered into the information of the eyeball. Same way with the fence. Points are down, pinks on this side, teals on this side. Upside down and backwards is how you actually see. 4.32, visual physiology. You have photoreceptors, which pretty much detect photons or light rays, and the visible light that you can see is from 400 to 700 nanometers. Basic unit of visible light is what a nanometer is. And photoreceptor structure pretty much is a visual pigments, and they're derived from a compound called rhodopsin. Bound to the pigment in the retinal area, and the retinal area, to help make this pigment, it synthesizes it from vitamin A. Figure 9.20, color vision. If you were colorblind, since these are all the same size and same object, and you would see variations of gray, you may not be able to see the 12 that is located in this visual test. This is a colorblind test. Bleaching and regeneration of visual pigments. So, photon comes in. This is the light ray, visible light. Retinal changes take shape. Bleaching separation. You get opsin, which is inactivated. Opsin comes back around, and it will actually retinal and opsin are resembled to form rhodopsin. This is how it makes this, so that it will pick up the photon, and this requires energy, or ATP. It's also, once the photon is assimilated and the bleaching separation, this separates off, ATP occurs, and retinal is restored so that it can pick up more information. Clinical note or visual acuity. Standard eye test is about at 20 feet, and the point of a standard eye test is to read the smallest letter. Variations of this as you look at it mean you can have 20/15, 20/10, but that is really good vision. Most people that do not have good vision, 20/40, 20/50, and so on, cannot actually see the smallest letter, so they would have to see larger letters on the actual visual acuity test. Clinical note here, accommodation problems, and these are myopia and hyperopia. Myopia is nearsightedness. Vision at close range normal, and the eyeball is pretty much too deep, so you have a problem focusing on the back wall of the eyeball. Hyperopia or farsightedness, and the eyeball is too shallow. You can see at distances clearly, so they call it nearsighted because you can see near and can't see far, and they call it farsighted because you can see far and can't see near. And this is normal right here. So myopia would focus, right there, and this would be nearsightedness. So when you're looking at things distance, it would be focusing it in the wrong location. If you have a corrective lens or glasses or a contact, this would refocus onto the right area and correct the myopia. Same way with hyperopia, which is farsightedness. Your eyeballs are set up to look at long distances. But whenever you have things at close intervals, it's at the wrong distance. Again, a corrective lens to refocus the image at the appropriate location. The visual pathways. The visual pathways begin at the photoreceptor and end at the visual cortex of the cerebral hemispheres. Now this is all the way in the back of your brain. So as we look at this, we have in a couple of slides we'll see this where it's actually assessed in the brain tissue itself. So from the back area, this is light coming in, bipolar cells receive the information, and these are rods and cones right here. This is the rhodopsin molecule, and these are on the discs that are in the rods here. And all this information is assessed and then brought into the cerebral hemispheres, and I'll show you where that is. So your right eye and your left eye kind of overlap in an area. Now this overlap is where you get binocular vision at. And that's how we normally see. You lose some of your periphery if you're only seeing out of one eye. So this eye here, left eye, will place its visual information in the right hemisphere and vice versa. Right eye in the left hemisphere. And this is something else noted from earlier, that this is assessed, and this sensory input is assessed and run through the hypothalamus. So all of this visual information upside down and backwards and retransposed in this cerebral hemisphere, upside down and backwards and retransposed into this cerebral hemisphere. 4.41 night blindness can occur because you are low on vitamin A. The body can convert carotene to vitamin A, and pretty much this is where we get that carrots are good for the eyes because they're chock full of carotene and vitamin A. Both. 4.42, we have various eye emergencies here that we're going to talk about. Conjunctival injuries, which is pretty much the coating on the eyeball. Corneal injuries, which would be lacerations and or abrasions. Blunt eye trauma, we might see some sticks hanging out of an eyeball in this. Chemical injuries, which would burn off or damage the cornea and conjunctiva. Ultraviolet keratitis, which is from what you would get from looking at welding for too long, and then acute glaucoma, which would be a spike or an increase in the pressure inside the vitreous humor. And these are conjunctival abrasions, trauma, or blunt trauma. Obviously down here at the bottom, this is pretty much blunt trauma. This would be an eyelid laceration or tear, and this would be conjunctival abrasion that occurred here and actually caused some hemorrhage on the sclera. Ultraviolet keratitis and acute glaucoma. This is the ultraviolet keratitis, which can be caused from looking at something that's too bright. You can get this from the sun, actually, by looking at the sun. And then this is acute glaucoma. This is on someone that already had some conjunctival problems to begin with, like cataracts. But do you see all of the blood vessels bursting? And this is not from actual conjunctival injury. All right, and that ends this section. This is finished of part 2 over chapter 9. If you have any questions, feel free to call me, 405-219-7613, or you can run me an email at smith@msa.net. Thank you.